Hi everyone, I'm Sally Kochmar, Lab Manager for the Magee Women's Hospital Med Genetics and Genomics Labs in Pittsburgh and Director of Education for AGT. And it's my pleasure to welcome you once again to AGT's webinar series. For those of you who are currently AGT members, this is renewal season. If you haven't processed your renewal, please do so as soon as you can. And for those of you who aren't members, I urge you to consider joining. For $95, it's a real bargain, and you get the journal of the association, which will give you free test yourself, continuing education, and it's the reason we can bring you these webinars, because those membership dues are what pays for the GoToWebinar series for us. So please join. I think you'll get a lot out of it, and it's an investment in your career. A few quick housekeeping notes before we get to today's presentation. Please feel free to ask questions anytime during the session. In the upper right side of your computer screen, there's a control panel, and in the lower portion of that panel, you can type in a question or comment and submit it to the webinar organizers, and you can do that any time during the presentation. At the end of the presentation, we'll take about 10 minutes to read the presenter the questions and have her answer them. If you are participating as a group, please designate one person in the group to complete the online evaluation. In order for everyone to receive their certificates of attendance, that person will need to enter the names and email addresses of each person who's participating at your site today into the evaluation. As a backup measure, you can also complete a sign-in form and fax it to AGT. Certificates will be emailed to participants approximately 30 days following the webinar. Today, Dr. Alexis Carter will discuss the basics of next-generation sequencing informatics. Alexis Carter is the physician informaticist for the laboratory at Children's Healthcare of Atlanta. She's the first chair of the new informatics subdivision in the Association of Molecular Pathology, and also serves as test directory editor and a member of the AMP Governing Board and Publications Committee. She's a past president of the Association of Pathology Informatics and is a member of the Informatics Committee and Clinical Informatics Steering Committee of the College of American Pathologists. She's immediate past chair of the International Pathology and Laboratory Medicine Special Interest Group of SNOMED CT International, which is the governing body for SNOMED CT terminology. She's a secretary for the Working Group on Two-Dimensional Barcoding for the Clinical and Laboratory Standards Institute, and is a section editor for informatics for Archives of Pathology and Laboratory Medicine, and is on the editorial board of the Journal of Pathology Informatics. She's a faculty member for the Clinical Informatics Board Review course presented through the American Medical Informatics Association. She's board certified in anatomic pathology, clinical pathology, molecular genetic pathology, and clinical informatics, and her clinical practice is both clinical informatics and molecular genetics pathology. Dr. Carter?
Okay. Hi, I'm Alexis Carter, and thank you for that very nice introduction. So I'm going to be talking to you today about next-generation sequencing basics of informatics. I don't have any financial conflicts of interest to disclose with regard to this presentation. By the end of this presentation, I'm hopeful that all of you will be able to understand the basics of next-generation sequencing and the importance of its informatics, to describe major file types and the quality metrics used in bioinformatics pipelines for next-generation sequencing, and also to explain the limitations of existing pipeline standards for file formats and validation. I'm also hoping that you come away with an accurate concept of what big data is, as well as a little bit about computational pathology, and also to understand the challenges and threats to NGS testing, including laboratory information system and electronic health record limitations for reporting. So there's been a lot of focus on next generation sequencing, and even more focus at the level of the federal government on next generation sequencing informatics. And there are a number of reasons for this. Result generation for next generation sequencing assays, unlike other laboratory assays, are absolutely dependent on very complex computational algorithms. The original sequencing reaction results are really not decipherable without them, and there's no visual or numeric quantitative analysis that you can go back to and look at to visually confirm the result that you're seeing. Next generation sequencing is very popular, both as being under scrutiny by the U.S. federal government, but also because of the power that we are now seeing with our ability to sequence so much of a patient's genome, whether that be in a tumor genome or in the patient's germline genome. So it's really all anybody ever talks about, it seems like nowadays. The other thing is that it does require utilization of big data resources for analysis in the vast majority of cases, so some discussion of big data is necessary as part of this talk. I'm sure many of you may have seen this picture before. This is actually from the federal government, NHGRI, and it compares Moore's law, which is not really a natural law, but basically a prediction that Gordon Moore came up with in 1965, saying that the number of transistors per unit area of integrated circuit, which is an example of which is a computer processor, will double every 2 years and often offered at decreasing cost. And I think that most of us who own computers or smartphones nowadays realize that we have so much more computational power available to us for a much cheaper cost than it used to be. Well, this diagram really shows that the cost per raw megabase of DNA sequence has, since about the late 2000s, far exceeded the predictions that were made by Moore's law for computers. We have seen a rapid decrease in the number of bases that we have been able to sequence per unit cost over time, and over the last 2 years we've seen another dip in that cost. And this is great for our patients because it means that we're going to be able to find out more about what's hiding in their genome for less money. Similarly, the cost per genome itself is following a similar trend, and so you can see that data there. So today versus what DNA testing was like when I first started doing it, which was a long time ago, previously, you know, we would have to test each gene, we would have to test each exon. It was very expensive to do. Sometimes it was very expensive just to do one gene. It was certainly expensive to do more than one gene, and we never even thought that we would be able to test the entire DNA. So today, there are multiple panels available out there. We can sequence many genes at one time or all of them with exome testing. Obviously, as we just discussed, the cost has really decreased and we can find more variants with less money. And all of that kind of relates to now we have a lot of variants that we don't necessarily know what they mean just yet. We're not sure if they're benign polymorphisms or if they're pathogenic variants. So what this means is that we have to start using big data resources to be able to accurately interpret this data and to make discoveries about genomes so that we're able to give the patient the information that they need. People use the term big data, and especially with how it relates to next generation sequencing a lot. However, most people don't really accurately use it, in my opinion. Big data actually does have a definition that goes with it. A lot of people think big data refers to next generation sequence generation. So that's 2 generations in one line, but what that means is just generating the sequence by itself because the files are very big, that is big data. And that is actually not the case. When you're generating large amounts of data, it actually doesn't satisfy the definition of what big data is. But there are aspects of the NGS analysis that do use big data resources, and we're going to talk about the difference between those two. So big data is really characterized by 3 V's. The first V is volume, and unfortunately it's volume that people usually think about being big data. But there are 2 other V's associated with big data that have to be present in order for you really to be able to call it big data. So for example, you have to have variety, meaning there are many different types of data coming into your data resource. And that means that in addition to sequence data, you would also have to have audio, video, text files, other types of data coming in that are of a wide variety of type. In addition to that, you also have to have velocity, meaning that your data resource is constantly accumulating new data all the time, and typically from multiple end users. Most of us, even if you're in a large NGS laboratory, probably don't satisfy those last two V's when you're talking about just generating the sequence out of your lab. So computational pathology, just to briefly delve into that, the specialty of computational pathology, which has been relatively recently defined by a large working group of people, the references for that will be at the back of this presentation, is really rooted in being able to analyze and use big data resources. So what many people don't realize is that a laboratory information system or an electronic health record, those actually do satisfy the three V's as being big data resources. And that is why computational pathology and other efforts are really looking at how do we utilize all of the data that's coming into those resources so that we can make better predictions about which patients are going to get sick, which patients may not, how can we better prevent disease in our patients. So, so when you're doing next generation sequencing, the FASTQ, the BAM, and the VCF files, which we'll talk more about later, they do have volume, but they lack velocity and variety. Unless you happen to be in a lab, and there are very few labs that actually satisfy this criteria, where you have multi-patient exome and genome level sequences that are acquired on an ongoing basis from multiple different types of analyzers, meaning you've got ion torrents, you've got Illuminas, you've got other NGS analyzers present in your lab, and they're all generating a variety of different sequences. However, when a pathologist or a geneticist or a laboratorian is actually going out to online data resources for variant databases to help them interpret variants, those variant databases frequently do satisfy the criteria of being big data resources. And we'll talk a little bit more about that here. So generating data in your next generation sequencing laboratory, the way that you've designed, that you've located that data, your data tends to be highly structured. There's very few people who are preparing that data compared to hundreds in a big data resource. And the longevity of that data, although some people would be horrified when they see short, but in some cases a small data resource is considered one of those where the longevity of the data is relatively short. In our laboratories, this would not satisfy that criteria. If you talk about online variant databases by comparison, and these would be things like ClinVar, My Cancer Genome, Cosmic, especially Cosmic, and some other databases, you really have a huge number of places that are actually generating that data. The data can be relatively unstructured because of the way that people are putting it in. Many people are actually contributing to those data resources, and it's not usually the person who's using the data, and the data stays there for a long time. Similarly, online variant databases compared to the data that's actually generated within your NGS lab may have different units of measure. They can be hard or impossible to repeat projects with new data because of that constant accumulation of new data. Obviously, maintaining some of these online variant databases is very expensive, and in some cases the data can be very loosely organized or not organized at all, which can also be challenging when you're trying to use data out of those resources. Again, that doesn't really impact our individual NGS labs in the vast majority of cases. Realizing all of this is relative when you're looking at comparing the data in your laboratory to what's in an online variant database. So next generation sequencing is the term that most of us use. The better term obviously is massively parallel sequencing. And this is where you're sequencing DNA in short overlapping fragments, and then you're aligning them to a reference and detecting variants. So this is a picture of the integrated genomics viewer, and I think many of you probably have seen this before, but when I'm interpreting NGS analysis, I really like to use this tool. It's open source and freely available. So you'll see here, you see the patient, the reference genome here, and each one of these gray lines is a read that is generated off of your NGS analyzer. The arrow tips at the end indicate the direction of the read, so you can see this one is a left to right read and this one is a right to left read based on the arrow tip points. In this case, here's the reference sequence. The reference sequence includes a cytosine in this location, and in this patient what we have is a thymine in almost all of the reads that are present. So this would indicate the patient has a variant in this particular location. So in NGS analysis, as many of you are probably familiar, you extract your DNA, or in some cases RNA. You do PCR of the selected regions if you're doing a gene panel with a targeted gene panel. You'll fragment your DNA, you'll ligate that DNA to adapters, and then you'll place that DNA on a flow cell or chip. And this is, as many of you probably realize, this is more particular to Illumina technology, but we will talk about ion torrent technology here in a minute. The adapters are placed, so you can see a picture here. So your adapters are placed at either end of the DNA fragments that you have off of your patient. And what you have are a flow cell binding sequence number 1, and then you have a flow cell binding sequence number 2. And these are different sequences because they want to know which end is which. Similarly, you have a sequence primer binding site, number 1 and number 2, and then you have indexes 1 and 2. So the adapter contains, so the flow cell binding sequence is the sequence with known complementarity to the binding site in your chip bead or on your flow cell oligonucleotide. The unique index that you see here is what they call a molecular barcode. For Illumina technologies, that's typically about 8 to 12 base pairs, but what that index does is for each one of your patients that you're going to be running on the analysis, you would give them a different set of indices. So you'll have, like, if you're going to run 5 patients on an analysis or on a flow cell, then what you would do is you would have indexes 1 through 10. Patient 1 would get index 1 and 2, patient number 2 would get index 3 and 4, and so on. This allows you to put multiple patients on a single chip or a flow cell, and that really helps to reduce the cost of sequencing. When I first learned about this, I will be honest, that really gave me the willies, but the technology is pretty good at separating these things out. So, and then you have your primer binding sites for the sequencing reaction. So delving a little bit more into Illumina technology, the flow cell has a lane which is coated with oligonucleotides which are complementary to those flow cell binding sequences in your adapter. The sample flows down the lane and it binds to the oligos that are present, and then you have sequencing by synthesis, which means that for every nucleotide it's attached to a different color, and there's a visual fluorescence recorded for each reaction for each nucleotide at those specific locations in the flow cell. The DNA strands then bridge over and bind to their other end to the flow cell, and the reaction repeats, and that's how you get both forward and reverse read sequencing. Ion torrent technology is a bit different. The beads are coated with oligos, which are complementary to the binding sequence. The DNA then binds to those beads, the DNA from your patient, that is. Amplification reaction occurs to coat the bead with identical sequences from your patient, and then the DNA-covered beads flow through a semiconductor chip and bind to wells in those chips, one bead in theory per well. You have single nucleotides, so for example you would have a wash of adenine nucleotides that wash over all the cells of the chip for about 15 seconds. And those cells which incorporate that base will release hydrogen, and then the pH is measured in each one of those wells, and it's determined whether the base was incorporated or not. After you've done your sequencing reaction, it's important to make sure that you have assigned those sequences that have been read to the correct patients. So demultiplexing is the process by which the sequences from those patients, when they come off of the instrument, all mixed together, start being separated by your computational algorithm by looking for the specific bases at the various ends, at the 2 ends of the sequence, to determine which sequences belong to which patients. And the computer code, obviously, which does this demultiplexing, must be robust. It must be part of your validation, and you have to have integrity checks to make sure that there's no overlap or mix-up of your patients as part of that process. So now getting really to the main part of this discussion, we're going to talk about the main components of a bioinformatics pipeline. So a bioinformatics pipeline, to be clear, means that you have multiple sets of one or more computational algorithms which are performed in a sequence or a series to analyze biological data. Bioinformatics pipelines are often discussed when you're only talking about NGS data, but a bioinformatics pipeline really can relate to any biological data, so you can actually hear of bioinformatics pipelines being used for proteomics, for example. It's really very important to collect and check quality metrics after each step of your pipeline. And there are many software packages out there which have variable quality and which may have variable sensitivity and specificity for certain types of variants that you're looking for in your patient's data. So going through this step by step, your NGS analyzer is going to generate raw sequence data. And then a computational algorithm will help perform the demultiplexing so that you're analyzing sequence from one patient at a time. Once that has happened, it will package that data into FASTQ files, which we'll talk a little bit more. So in the FASTQ file, what that contains are sequences, the sequence reads that you got off of the instrument. After you have the FASTQ file, you'll have computational algorithms which align those sequences to a reference genome and which generate a SAM file, which can then be further compressed to a BAM file. And we will be discussing each one of these file types here in a moment. So your BAM file is then generated, and after you have a BAM file, there's another set of computations run on the BAM file to detect variants from the reference. So it's looking at where the patient's data has aligned to the reference genome and is then determining where there are variants from that reference. And once that has been determined, another file format called a VCF file gets generated, and then the pathologist gets the VCF file, will perform any annotation or will have the first round of filtering actually for anything that might be an artifact or other things like that, or known benign polymorphisms. Those will be filtered out, and then the pathologist or geneticist will be able to do the interpretation off of the annotated or filtered VCF file. So a FASTQ file format, we'll first start with what's the difference between a FASTQ and a FASTA or FASTA file format. A FASTA file format is a simple text file format which has nucleic acid sequence. There's really no well-defined or accepted standard for how a FASTA file format is constructed. By contrast, a FASTQ file format is a FASTA file format, but it has additional quality data for each base that is in the sequence, meaning how valid is that base call for each position in the sequence. Also, the FASTQ file format really also doesn't have an internationally defined or accepted standard as to how it's constructed. It's considered a de facto standard because so many of us use it. And the one that most people use was developed around the year 2000 by the Wellcome Sanger Trust in London, and I have the reference down there for you. We're going to go into a little more detail on the FASTQ file. The FASTQ file incorporates what's called a FRED or a Q-score for each base call. So there are 3 versions of a Q-score, and none of them are very well defined. There's one from Sanger, Alexa, and Illumina, so I just picked Sanger since that also seems to be the one that most people tend to use. So basically a Q-score, what it does is it takes, it performs this calculation based off of the probability that the base call at that particular position is erroneous. So if you have a chance of 1 in 10 or 10% that the base at that location is incorrect, your Q-score will be 10. If the chance is 1% or 1 in 100 that you have a wrong base in that location, your Q-score will be 20. If you have a base score of 0.1% chance that it could be incorrect, or 1 in 1,000, then that would give you a Q-score of 30. In most of the labs that I've dealt with, we really like to see Q-scores for each individual base to be at least 30 or higher to make sure that we're calling the right thing, because if we have even just one base that's incorrectly called and we call that on a report, then we could be giving a patient a false positive diagnosis. So this is actually what the FASTQ files look like. FASTQ files, as well as the other file types that we'll be discussing, really were not developed for humans to read. They were developed for computers to read. So these files are extremely computer parsable. The first line of a FASTQ file always starts with an @ symbol, and it has a sequence identifier and an optional description. So really the formatting of this line is almost entirely up to the laboratory. There's actually no requirement to include the sample identification as part of this. I hope that horrifies most of you because it horrifies me. Most laboratories do take care of this on their own, but I have seen some very interesting practices in some laboratories as to how they actually identify samples within their FASTQ files, and we can talk more about that potentially later. The second line of the FASTQ file has the actual raw sequence, so this should look familiar to many of you. So this was the sequence that was read off. The third line may be blank, but sometimes it optionally will repeat the sequence identifier. So this is the identifier that comes off of the NGS instrument, and it may repeat the description that the laboratory has decided to put in it. The fourth line is where this really differs from the FASTA format in that for each base that you have in line number 2, you're going to have a corresponding letter or symbol in the fourth line of the FASTQ file. And we'll look at this more in a minute, but these indicate the quality at each location. Now I know you're going to be confused by that because I just said that the Phred scores were 10, 20, 30 and like that, but hopefully this next slide will help that make sense. Basically, in order to save space and because we don't want to be repeating numeric characters that can run the risk of being one off and then causing problems, what they did is came up with a conversion table so that if your Q-score, for example, is 30, you actually would see this symbol in this line. In this particular case, what we see is that the first base in this FASTQ sequence, this first guanine, has a Q-score of I. If you go over here, that I will relate to a number of 41, which is a very good Q-score. So in most cases, for these bases, they had a very good Q-score assigned to each one of those. This would correlate with a greater than 1 in 10,000 probability of error. However, you'll notice that we did have 2 bases in this sequence that got a Q-score that was converted to a 9. That would be a Q-score of 25, which is less than what most of us like to have in our NGS laboratories for the probability of error. Moving on to the next file format type, which is the SAM file or BAM file, depending on what you use in your laboratory. SAM stands for the Sequence Alignment Map. It also, like the FASTQ file, is a free text file. It contains data showing where the sequence in the FASTQ aligned to the reference sequence. Most of us are still using the Human Genome 19 reference sequence from 2009, but there are some laboratories that may be using the Human Genome 38 or GRCH 38 sequence from 2013. The SAM file does have some more structure than the FASTQ file. We're not going to go into terrible detail there, but again, when you look at the SAM standard, which I'll give you the reference to in the next slide, there's really no requirement or standard for sample identification in a SAM file. So here's the link to the SAM file standard, and you can see that there are multiple lines, and here's where you start with the alignment data. Again, these were designed for computers to read, not for humans to read, but you can go through the standard and find out for each one of these columns, for example, what that data item is supposed to represent. The BAM file, which is what many of us use, is basically a binary version of SAM, which means it's compressed in this particular case. So it takes up less memory size than a SAM file. And it allows good compression of the SAM file, and some of these SAM files are very large, and it still allows us to sort of randomly interrogate those files for the specific alignment sequences that we're looking for. So the BAM file is more commonly used because it has this lower file size and you can still access the data randomly like you need to. And then the VCF file is the variant call format file. This is the text file that contains a list of variants from the reference, and it can include, when it is initially generated, all variants that are seen, including artifacts that may be a part of your NGS analysis, artifacts being false positives in the sequence that may be seen over and over again with multiple patients. It can also include benign polymorphisms, as well as variants of undetermined significance and pathogenic variants. Again, the VCF file format standard does have a de facto standard, which I've listed here, but there is no requirement for stringency or for sample identification. And there are multiple versions of this variant call format file in use. So the VCF file, this is what a VCF file looks like. The initial part of a VCF file has a huge number of data elements that you can put into it, and some of this is kind of up to you, but then you start listing the actual variants down here, again meant for computers to read and not for humans to read, but if you read the standard, it's fairly easy to determine what some of this is. So for example, this is the chromosome number, this is the actual coordinate where the variant is. This is an ID, which can vary between labs, but many of you will recognize that this is a dbSNP identifier. The reference nucleotide was a guanine, but the patient actually had an adenine at that location. This is the Fred score, the quality score, so 29. It passed the NGS analyzer's filter checks, and then there's additional information here that you can see. Some of this is laboratory dependent. So I did want to let you know that the Centers for Disease Control and Prevention have facilitated a working group starting in 2012, and they are trying to develop a clinical grade VCF format. And I think that this is very exciting. I actually talked to one of my colleagues who has been driving that particular working group just yesterday, and he told me that they are in the very late stages of getting this out for its first publication, so it'll be very exciting to see that come out. And the goal really is to identify and build consensus around what needs to be included in a clinical grade variant call file format. And more information about it is at this website. So a few other things about NGS sequence data which may be already known to most of you. A read is basically a single output sequence from an NGS sequencing reaction, and depth of coverage are the number of reads which contain a specific nucleotide at a specific location. Most people, when they're reporting depth of coverage, are reporting their average depth of coverage for the analysis that they're performing, and depth of coverage is very different depending on the type of testing that you're doing. A lot of germline or inherited testing, because your sequence variant allele burden is usually either going to be 0 because they're normal, 50% because they're heterozygous, or 100% if they're homozygous, their depth of coverage may be lower because it's easier to pick up variants when you're looking at germline testing. However, with somatic or acquired testing, such as variants that you might see in tumors, the variant allele burden can be quite variable depending on the sample that you're looking at, because in many cases it may be contaminated with normal, or you may have significant intratumoral variability, meaning that some pieces of the tumor have a variant and some don't. It's typical in most labs, although I've seen this down as low as 3%, to make sure that you have at least 5% allele burden before you'll actually make a call about a variant in a tumor. And the depth of coverage, for this reason, because our percentages have to be able to go so much lower for detection, our depth of coverage needs to be a lot higher to be able to catch that. Most of us will use 500x or higher. I personally like to see 1,000 to 2,000. That's my what makes me feel a little more comfortable when I'm looking at these assays. So there are many computational algorithms that are involved in even just a single pipeline. And because some pipelines actually can be set up to filter data, meaning to hide certain variants from you based on certain predefined criteria, you have to be very careful to make sure that those filtering criteria are not improperly designed, because if they aren't properly designed and validated, it can actually cause real variants to be hidden from view. So I'm going to give you one example of that. A laboratory that I know was notified of a discrepant result. I have to say it's not the laboratory I work in currently. So it was notified of a discrepant result, and what happened was there was a cancer sample that was analyzed at another lab on the same patient that had been analyzed in the lab that had been called, where there was a 15 base pair insertion in EGFR. And for this particular tumor, that was a clinically significant variant that was detected at the outside lab, but the original lab had called that negative. So the original lab pulled out their NGS data and reanalyzed it. And what they determined was that the original pipeline had been built to exclude a variant if there were 3 or more unrelated variants that occurred at the same location. So regardless of the percentages of those variants. So for example, if 1% of the variants at a particular location were artifacts, but there were 2 different artifacts in the same location, and then you had 15% of your base pair, 15% of your alleles had a 15 base pair insertion in exon 19 of EGFR, then this pipeline was designed to throw all of those variants out. And so the pathologist who had signed out this case actually had no knowledge that this variant existed because it didn't show up in the VCF file that was presented to the pathologist for initial analysis. So obviously this is a very scary thing. None of us want us to have this happening in our laboratories. So this case is being shown to you to illustrate the importance of going through validation and making sure that you fully understand what is built into your pipelines. So a few more words about NGS analysis. So interpretation is the process by which you're assigning clinical significance to a variant. And usually that's going to be done by an advanced laboratory professional, such as a PhD, a physician, or a pathologist, or a geneticist. It can occur with or without the assistance of other validated tools to help you with that. And your basic interpretations for a variant are going to be that it's an artifact, meaning it's a false positive that's generated by the sequencing process. It can be a benign polymorphism. It could be a non-coding and synonymous variant. It could be a known pathogenic variant or a variant of undetermined significance. So annotation is slightly different. It's the labeling of a variant in the context of a particular clinical presentation, specifically for future use in the analysis of other samples. So for example, a BRAF V600E has very different clinical implications if it occurs in a pediatric brain tumor versus a melanoma versus a lung cancer versus a colon cancer. So annotation would be the process by which you're assigning the significance of that variant within the particular way that it was presented. Was it in a melanoma, for example, or did it show up in a pediatric brain tumor? It also allows you to potentially use your information systems to link that variant to specific online databases, and it also the unfortunate thing is that most laboratories kind of lack adequate tools to really help them annotate variants and to retrieve those annotations for future analysis on other patients. So in my experience, it depends on the analysis that you're doing, but only about 20% of the variants that you find in a next generation sequencing assay have known significance, meaning they're either known pathogenic or known polymorphisms or artifacts. The other 80%, in my experience, have to be researched to some extent. And this can be a very painful process, especially if you're trying to be careful, where you're basically looking in a lot of different online variant databases to help you determine what the sequence of that variant is. Do we keep it as unknown, or do I find data in these online databases which indicate that it might be pathogenic or it might be a benign polymorphism. Each of these online databases that you see here, and there are many more, are constantly being updated by multiple and often anonymous sources. The data can be somewhat unstructured, particularly when you're talking about PubMed. It can get really interesting when you're trying to find papers for a specific variant, especially given the heterogeneity with which some variants have been named. And the data is often uncurated. There are some of these databases which are very highly curated and others which aren't really curated at all. And when you start looking at all of this, you realize this is really the definition of big data, and this is the big data component of looking at NGS assays. So just to give you an example of what this means then, this is an example of doing big data analysis. Each one of these data points is a predictive model about where a hurricane might go. So this was Hurricane Sandy in this case. So needless to say, this is very hard for humans to sort of filter through. The yellow lines and these other lines here actually are talking about making bigger predictive models based off of the numerous little ones. And so this is something that's actually much easier for humans to look at. Similarly, when you have a small data resource where you've got sort of very defined data up front, you use a few tools, you use some knowledge, and you're able to come up with something hopefully actionable. But when you're dealing with big data, such as these big data resources, there's a lot of noise, which would be represented by weeds in this particular photograph. So in order to really be able to use big data resources, you have to apply some tools and knowledge to be able to get them into a state where you can actually use the data for what you need to use it for to get actionable variants out of that. And this is really where big data analysis resources are focusing. So for example, many of you may have heard of IBM Watson Genomics, which is, you know, the Watson from Jeopardy, that was using all of its harnessing power and artificial intelligence to be able to beat two of the Jeopardy players, the biggest winning and the highest number of game-winning Jeopardy champions. But they've now turned Watson to healthcare. And so what they're using Watson for is much of what I'm talking about in this slide. So when you're looking at these online databases to do your interpretation, there's a lot of genomic, what I would call noise in these databases. So you have a lot of variants which are reported as being associated with disease when really they're a benign SNP, or they haven't excluded the possibility of a benign SNP, and vice versa. And you have to remember that the Human Genome Reference, or HG19, as well as HG38, those are a Caucasian male, and since I'm certainly not male, and many people I know are not Caucasian, comparing genotypes to a Caucasian male is going to generate a lot of variance from that reference, which may all be benign polymorphisms. So it's very important to be able to look at this data and be very careful about how you're analyzing it. Often the amount of noise, in my experience, is proportional to the amount of data. So in a database like My Cancer Genome, this doesn't have a huge amount of data, and it is very highly curated. So generally speaking, a variant doesn't show up in My Cancer Genome unless it really does have some implication for the tumor. If it doesn't have an implication, it says so up front. Versus Cosmic, where I've actually found multiple benign polymorphisms listed in Cosmic for various tumors. So you always have to examine the data, I think, from multiple resources in order to be able to correctly interpret it, and also to be able to correctly interpret it in the context in which it shows up, whether it's germline versus acquired, what tissue is it in, what tumor type, and what is the patient's phenotype. So, and I think many of you are probably aware of the FDA LDT draft guidance. The exciting news about that is that on November 18th, just recently, the FDA decided not to pursue this. However, the FDA did have very good reasons for the concern over how NGS assays are being performed, and I do not think the FDA is going to drop this particular issue just because there has been an outcry from patients about the safety of some of these tests that are available. So I think a lot of organizations, including the Association for Molecular Pathology and others, are going to be looking at how do we provide standards and guidance to make this type of testing safer, especially given how difficult it can be to pull the data together and interpret it based on the last slide. So these are the references, some additional references in addition to the ones that I gave you throughout the talk. And at this point, I'm happy to answer any questions.
Thank you, Dr. Carter, for your excellent presentation. Just a reminder to any of the participants, if you have a question, please type them in on your webinar's control panel so we can get them to Dr. Carter. And as are also reminding everyone that the webinar participants will get an email from AGT that includes a link to the evaluation, and please access it and provide your feedback on the webinar session today. It was recorded and it will be posted on the AGT website very shortly, and it will be available with a quiz for those colleagues of yours who were not able to attend today can access the webinar and the quiz to get the CEUs for it. Okay, I think I have a question coming in here. Will there be, they want to know if there's going to be copies of your PowerPoint.
Yes, I did submit a PDF of the PowerPoint to AGT, so they should have that.
Okay, great. Please email AGT for that. Do you think that NGS data analysis will ever be a task that lab technologists will be asked to perform instead of high-level PhDs?
You know, I don't know. You know, when I started my fellowship, if you had ever told me that I was going to be signing out an assay that had 150 genes in it at one time, I would have told you you were absolutely crazy. So I may not be the one to ask to predict. For right now, and for the foreseeable future, because there is so much interpretation that has to occur for these variants, and because we still are only at the tip of the iceberg as to what we actually know about what these variants mean. And it's not just, you know, a single variant within a tumor. As you know, there are different variants that can occur which interact with each other, and can one variant can actually mitigate or promulgate the pathogenicity of another. And so, you know, I can't, at some point we could see computers doing most of this, but I don't think that that's going to be any time in the near future. But again, I would have told you 10 years ago, there's no way we're going to be doing exome sequencing, you know, during my career, and I have been totally proven wrong on that. So I always have to be cautious about trying to predict the future.
Is there training out there for people that want to go into this field of bioinformatics? Do you think there's adequate training being offered by universities?
You know, I'm really glad that question was asked. There is no, to my knowledge, formal training. So let me take a step back. There's lots of training out there in biomedical informatics and in bioinformatics in PhD programs and in postdoc programs. However, unlike, for example, the American Association of Clinical Chemistry, I'm sorry, that's the wrong thing. The American Board of Clinical Chemistry offers a board exam for clinical chemistry PhDs who have done a fellowship to be able to be clinical laboratory medical directors. Same thing for microbiology and several other specialties. We don't have an equivalent board certification or fellowship training program for PhDs who have been trained in bioinformatics because, as many of you know, doing bioinformatics in a research laboratory is a very different animal than doing bioinformatics in a clinical laboratory. Frequently the PhDs that I talk to who have been in a research lab and are now in a clinical environment are shocked when they find out that they can't change a line of code from one assay to the next without doing validation all over again. So There have been discussions within some organizations about whether or not the organization should pursue trying to offer a fellowship and board certification for this type of training. I think those discussions are very early, so there is no formal clinical training for bioinformaticists, but there is a lot of PhD level and postdoc level training in bioinformatics, which may not necessarily be clinically based.
Great, thank you very much. At this time, I don't see any other questions, so we're going to just end the webinar for today. We'd like you to join us for upcoming webinars over the next several months. We'll be sending out a notification to members once registration is available for our next webinar, which will be in January, I believe on, it's a biochemical based webinar. We hope to see all of you at AGT's 42nd annual meeting in St. Louis, Missouri, which is scheduled for June 15th to 17th. Registration will be open in early 2017, and I believe the call for abstracts has already gone out, so please check your email for that. Thank you very much for attending today.